Good morning. I would like to welcome everyone to today's MD Anderson Cancer Center Enjoy Science seminar. This is a series of lectures given by MD Anderson faculty members and is organized by the Office of the Chief Scientific Officer, Dr. Giulio Draetta. I'm Alessandra Ferraioli and have the pleasure of hosting this series with my colleague, Dr. George Kalin. The goal of the Enjoy Science initiative is to strengthen the connection between the scientific and medical community at MD Anderson and our colleagues, our patients, and our friends around the world during this time of limited in-person at the bench and in the clinic activities. Today's seminar is being recorded and will be made available on the Enjoy Science website later this week. The speaker of today is Dr. Anna Aparicio. Dr. Aparicio is an associate professor in the Department of Genitourinary Medical Oncology, Division of Cancer Medicine at the University of Texas MD Anderson Cancer Center. Dr. Aparicio is an expert in the field of prostate cancer and has received numerous awards and recognition for her work. She has extensively published on the clinical characteristics and of treatment of aggressive prostate cancer. Therefore, Dr. Aparicio will be discussing for us the interesting topic of aggressive variant prostate cancer, what they are and why they matter. Thank you.
Thank you very much for the kind introduction and for inviting me to speak in this seminar, which I think is a fantastic idea. I've enjoyed all of the previous ones. So I'm going to talk about aggressive variant prostate cancers and try to give you a perspective of why these are important tumors to take into account. So these are my disclosures. So the bottom line, the take-home message of this talk is that prostate cancer is a heterogeneous disease. I often hear or read grants or papers about prostate cancer that discuss prostate cancer as if it were all one disease, a homogeneous entity. And much like breast cancers, where we know there are different subsets, biological subsets that have therapeutic relevance, or leukemias, which you distinguish chronic from acute, and then with them various subtypes, or any of the other malignancies for which we've arrived at molecular classifications that are therapeutically relevant. The same is true for prostate cancer. Our limitation is that our molecular classification is not quite there, and that's what we're trying to arrive at. So the dogma is that prostate cancer is essentially a disease of the androgen receptor. The androgen receptor, as many of you know, is a nuclear hormone receptor. It has an N-terminus domain, a DNA binding domain, and a ligand binding domain. And it normally lives in the cytoplasm bound to heat shock proteins. Upon entry of its ligand, testosterone, which gets converted to dihydrotestosterone in the cancer cell, the androgen receptor gets released and enters the nucleus, where it sets off a program of cell proliferation and survival that basically drives prostate cancer. So for this reason, the mainstay of prostate cancer therapy is that of androgen signaling inhibition. And we have various ways of achieving this androgen signaling inhibition, either by ligand depletion or through direct inhibitors of the ligand binding domain. And this was actually the first treatment that was discovered for prostate cancer in 1940. Many of you know it warranted a Nobel Prize. And basically, what was shown was that if you decrease or suppress the testicular androgens through orchiectomy, and more recently with LHRH agonists or antagonists, or if you also suppress the adrenal or intratumoral androgens with other more recent drugs such as abiraterone, you arrive at a tumor response. So the vast majority of prostate cancers, when you subject them to these ligand depletions, will shrink and will stop growing. And the same is true of the AR ligand binding domain inhibitors. These are drugs that have been around for a long time, but more recently we have what are called the second generation AR ligand binding domain inhibitors, which include more potent drugs such as enzalutamide, apalutamide, and darolutamide. Now, what we've also known now for a very long time is that although the vast majority of prostate cancers will respond to some degree to androgen signaling inhibitors, the fact is that the response to these drugs is extraordinarily variable. So this is a Kaplan-Meier curve that's shown of a clinical trial that was conducted in the last century. In which over 1,200 patients were randomized to receive either an LHRH agonist to suppress the testicular androgens alone, or this LHRH agonist together with, at the time, the available AR ligand binding inhibitor, flutamide. And you can see there was really no difference. This was a weak inhibitor. More recently, there does appear to be an improvement in outcome when you add more potent secondary androgen receptor ligand binding inhibitors. However, what this chart shows you is that while the vast majority of men had a median survival of 5 years, there's still substantial survival beyond 5 years and even after 10 years. And that's true, and probably even truer today with our more potent signaling inhibitors. But what that also tells you is that there's a proportion of men that have a very poor response to these androgen signaling inhibitors, progress and actually die within 1 year or 2 years of their diagnosis. And so what explains this poor outcome if the androgen receptor is truly central to this disease? Well, various mechanisms have been proposed throughout the years, and they can basically be classified into 3 broad groups. There's restored AR signaling. We've seen that there AR ligand binding domain mutations emerge that explain the resistance to these agents. There are androgen receptor splice variants that may also explain this resistance. And as I alluded to earlier, there's a synthesis of adrenal androgens and intratumoral androgens that can drive the AR program. There's also some work more recently that suggests that different hormone receptors, such as the glucocorticoid receptor, will bypass the need for the AR. But what I want to focus on is this group of androgen indifference, where basically it seems like the AR is not really playing such a prominent role in their progression. And the question is, are these primary or are these secondary mechanisms of resistance? So I think what our work and that of others has shown is that this program of androgen indifference may explain a substantial proportion of those that are primarily resistant to androgen signaling inhibitors, although some of them may also be explained by upfront excess of adrenal androgens and intratumoral androgens. And then there are secondary mechanisms of resistance that come in play, as I described earlier, and it is likely that the secondary androgen indifference mechanisms are overlapping with those of the primary resistance. The problem that we have is that although these androgen-indifferent tumors are responsible for a large majority of the prostate cancer deaths, we do not really have any therapies that are specific to the androgen-indifferent tumors. I've listed here the drugs that I mentioned earlier, and it's a fairly long list. And I've listed on the right side drugs that are not presumed to mediate their effect through the androgen axis. But none of these drugs are actually specific to the androgen indifference tumors. And why is that? Well, the problem has been that we have no way of recognizing the androgen indifferent tumors. So there, if you triple negative breast cancers, you can recognize them and you can set them apart and do a clinical trial and preclinical work that is specific to that subset of the disease. We don't have this in prostate cancer. And so what happens is that a patient that basically looks similar when they walk in your clinic with metastatic prostate cancer, you will start them on an androgen signaling inhibitor, but you have no way of predicting whether they will be alive and well 5 years later, or whether they will succumb to their disease within a year of their diagnosis. And so there's an urgent need for us to identify biomarkers that can allow us to enrich for these androgen-indifferent prostate cancers and enable the development of effective therapies for this subset. So we and others turned to a rare histological variant of prostate cancer that's called a small cell or neuroendocrine prostate carcinomas. I'm going to take a minute here, even though it's not on the slide, to make the distinction between prostate cancers with neuroendocrine differentiation and small cell or neuroendocrine prostate carcinomas. They are not the same thing. So you can have a garden variety prostate cancer that expresses neuroendocrine markers such as chromogranin A or synaptophysin that responds just fine to androgen signaling inhibitors. It is true that small cell or neuroendocrine prostate carcinomas often express neuroendocrine markers as well, but not exclusively. So the expression of neuroendocrine markers is not exclusive of the it's not specific for the neuroendocrine prostate carcinomas. And that's very important because the presence of small cell or neuroendocrine prostate carcinoma morphology has therapeutic implications. Why? Because they are, for the most part, AR negative. They do not express the androgen receptor. And thus they do not respond to androgen signaling inhibitors. They're often associated with atypical clinical features that I will describe shortly, but they are sensitive to platinum-based chemotherapies, just as small cells from other from other organs. Now, this is a rare event at initial diagnosis. Less than 1% of prostate cancers present with small cell or neuroendocrine prostate carcinoma morphology. But this morphology may emerge during castration resistance. And when it's present, it predicts for a virulent clinical course and a dismal prognosis. Again, we don't have good therapies for this disease. And we and others have done a lot of work to show that indeed the neuroendocrine prostate carcinomas are molecularly distinct from typical adenocarcinomas. So they have greater number of copy number alterations, they have distinct epigenetic profiles in terms of DNA, and this has been shown for DNA methylation profiles. They have distinct gene expression profiles. So as you would expect, since they are AR negative, they have decreased expression of AR and luminal epithelial differentiation markers. They have an increase in the expression of proneural development markers and stem cell-like markers. And they also express aberrant DNA repair and cell cycle gene pathway genes. As in addition, as with other small cells of other organs, they frequently harbor p53 mutations or deletions, RB loss, and PTEN loss, and these are often in combination. So we were interested, well, or there was an interest at MD Anderson a long time ago. Dr. Logothetis conducted this clinical trial to see whether the addition of doxorubicin would improve this dismal prognosis of the small cell prostate cancers. What they did is, this was a phase 2 study. It was a single institution, so obviously they tried very hard to find small cell morphologies. And so they biopsied anybody that would walk in the door that had one of the atypical features that is often associated with small cell cancer morphology. And what they found was that more often than not, instead of small cell cancer morphology, at the end of that needle, they would find a variety of different morphologies, ranging from typical adenocarcinoma morphology, poorly differentiated morphologies, large cell neuroendocrine morphologies, and of course, small cell cancers on occasion. But the point was that these were heterogeneous morphologies that all came about with atypical and virulent clinical presentations. So, as I stated, these heterogeneous morphologies were associated with the atypical and virulent clinical features of the neuroendocrine prostate cancers. And so that begged the questions, well, do the clinical features alone, even if we don't prove that there's neuroendocrine prostate carcinoma morphology at the end of that biopsy, predict for shared chemotherapy sensitivity? In other words, if someone walks in with these atypical clinical features, should I be giving them platinum-based chemotherapy instead of androgen signaling inhibitors? Will they do better? And this was a very important question, because now we're a little more comfortable giving chemotherapy, but back in the day, if you gave chemotherapy to somebody with prostate cancer, you were not looked at well. And on top of that, if you tried to give them platinum-based chemotherapy, which was considered very aggressive at the time, that was considered almost a sin. So this was a very important clinical question. So what we did is we compiled a set of clinical pathological criteria that are often associated with neuroendocrine prostate cancer morphology. Of course, if you have the morphology, that's one, but the presence of exclusive visceral metastasis, which rarely happens in typical prostate cancer, the presence of lytic bone metastasis, which is distinct from that of the typical blastic bone-forming metastasis of prostate cancer, the presence of very bulky tumor masses, including very bulky primary prostate tumor masses. The expression of low PSA levels relative to tumor volume. Generally, PSA is somewhat associated with the volume of tumor in a patient. But in this particular type of disease, they express low levels of PSA despite having a large volume of disease. The presence of neuroendocrine markers, but not alone, with high levels of serum CEA or LDH, indicative of poor differentiation, and primary castration resistance, which basically we defined as a less than 6-month response to upfront hormone deprivation therapy. And so we conducted a clinical trial in which we selected men that had at least one of those 7 criteria to give them, to treat them with docetaxel and carboplatin, and then upon progression, the etoposide and cisplatin, since that was the standard therapy for small cell prostate cancers. And so indeed we showed that there was a high response to the carboplatin and docetaxel chemotherapy in these patients, much as you would expect had they all had neuroendocrine prostate cancer or small cell cancer morphology. So then the next question we wanted to ask is, okay, so they share the clinical features, they share the chemotherapy response profile. do they share the molecular profile of the small cell prostate cancers? And so through a lot of work that I'm not going to detail in the interest of time, but basically immunohistochemistry and copy number analysis of tumor samples from participants in this clinical trial, as well as correlative studies from in patient tumor-derived xenografts from actually patients that participated in this particular clinical trial. Through all of this work, we arrived at this molecular profile that characterizes the clinically defined aggressive variant prostate cancers. And basically, that consisted of combined defects in at least 2 of these 3 tumor suppressors, so P53, RB, and PTEN. And we actually gained confidence in this molecular profile because preclinical models of androgen indifference are actually characterized by this profile as well. So in genetically engineered mouse models, the loss of P53 alone, the loss of RB alone, or the loss of PTEN alone do not result in androgen indifferent disease. However, when you lose 2 out of those 3, you have an increase in EZH2 and stem cell markers such as SOX2, and you have the emergence of these androgen indifferent tumors, often with neuroendocrine prostate cancer morphology, although not always. Okay, so we have done a single-arm clinical trial, and we have treated men with the combination chemotherapy up front. And the question was, well, is this really platinum, or is it just that it's just the taxane alone? And Peter Thal did a very elegant comparison to a previous looking at prognostic markers from a previous taxane-only clinical trial, and showed that indeed the observed overall survival from the patients that participated in our single-arm phase 2 study was greater than that of the expected overall survival when we used these predictors. And so Paul Korn in our group designed this clinical trial of cabazitaxel, a second-generation taxane, plus or minus carboplatin for the treatment of men with metastatic castration-resistant prostate cancer. And what he did is he stratified for the presence or absence of these aggressive variant clinical-pathologic criteria. At the time, we hadn't yet defined the aggressive variant molecular profile. And what he showed was that indeed carboplatin added to cabazitaxel improved the median progression-free survival of men with metastatic castrate-resistant prostate cancer by a tiny little bit, as you can see there. We then looked at demonstrated, that the presence of aggressive variant clinical pathologic criteria, which patients were stratified by, did indeed enrich for benefit from the carboplatin. How about the aggressive variant molecular profile? Well, we retrospectively, in a post-hoc analysis, looked at the available tumor samples by immunohistochemistry for P53, RB, and PTEN, and also in collaboration with Nicholas Navin, we did whole genome sequencing of the circulating tumor DNA to identify this signature. And as you can see, approximately half of the patients had the clinico-pathological criteria, and approximately half of the patients, a little less, had the molecular signature, although the numbers were somewhat small. What we found, again, through a lot of work, was that if you had the aggressive variant molecular profile by either IHC, immunohistochemistry, and or circulating tumor DNA analysis, you had significant benefit from the addition of carboplatin to cabazitaxel. So as you can see on the graph on the left, In the red are the patients that received the combination of cabazitaxel and carboplatin. In the black line are the patients that received cabazitaxel alone. And there was a significant difference in overall survival for those that had this aggressive variant molecular profile. Whereas those that did not only did they not benefit from the addition of carboplatin, but it appeared that there may be a trend towards a worsening, whether that is biology or simply toxicity of the combined therapy in the absence of benefit, or it remains to be determined. Paul Korn is going to lead a SWOG phase 3 trial in which we will now stratify patients by the presence or absence of this aggressive variant molecular profile to confirm these results. So I hope that I've convinced you that the aggressive variant prostate cancers are a clinically and biologically distinct subset of prostate cancers with unique therapeutic vulnerabilities. But of course, there's a lot of work that remains to be done. I've walked you through how we came about the aggressive variant clinicopathological criteria, the immunohistochemistry, the aggressive variant molecular profile by immunohistochemistry, and that of the aggressive variant molecular signature by genomic sequencing. So I did not specify that what we called aberrations in P53RB and P10 genomically are listed down here. So this included a wide variety of different alterations. And of course, it should be noted that for P53, while missense mutations that increase the stability of the protein are detectable by immunohistochemistry, of course, deletions, which is what was modeled in the preclinical studies, deletions do not, in fact, are not, in fact, detectable by immunohistochemistry. And that, of course, is compiled upon the issue that not all gene mutations are in one gene, obviously, are equivalent. And this has become obvious to many people in other tumor types, but in prostate cancer, we seem to have some resistance to that notion. Except for recently, these very two elegant papers showed that indeed distinct sort of genomic alterations in this gene, FOXA1, which are very prevalent in prostate cancer, will have very different phenotypic outputs. And so a big piece of what we're doing right now is trying to understand that distance from the aggressive variant prostate cancer molecular profile to the aggressive variant or androgen indifferent phenotype. And the other thing that of course, remains to be done is to link this aggressive variant molecular profile to clinical androgen indifference. So we have done this in preclinical models, but in clinical trials, we have yet to link the aggressive variant molecular profile to clinical androgen indifference. And to do this, we have taken advantage of a clinical trial that was recently completed called the Dynamo clinical trial, in which we treat men with metastatic castrate-resistant prostate cancer with maximal androgen signaling inhibitors, abiraterone and apalutamide. And based on their PSA decline in circulating tumor cells, at the end of 8 weeks, they get allocated into one or another group. If their decline is satisfactory, they get randomized to the addition or not of ipilimumab. Because the hypothesis is that the androgen signaling inhibitor sensitive tumors that are more likely to respond to immune checkpoint blockade, whereas those that have the unsatisfactory serum decline will have cabazitaxel and carboplatin and added to the androgen signaling inhibitors. And we were very fortunate. Our patients were very generous and donated a large number of metastatic tumor biopsies that are being subject to immunohistochemistry for the aggressive variant prostate cancer markers, and have also been subject to bulk RNA sequencing. And so these is some of the preliminary data that we have obtained from these tumors, which this is an initial set of the RNA sequencing. We are COVID sadly delayed the completion of these studies. Hopefully we'll get back soon. But basically what you can see is that the RNA sequencing profiles of these tumors can be in unsupervised hierarchical clustering results in two distinct clusters, and the top GSEA enriched pathway is the androgen response pathway, which indicates that these transcriptional profiles can indeed distinguish androgen responsive from androgen indifferent tumors, with the cluster on the right being enriched for EMT pathways, which is again has been associated with androgen indifferent tumors. And another question, of course, that we have is how do we link these aggressive variant prostate cancers that we defined starting from the clinic as a unique subset to the molecular profiles or the genomic profiles that are emerging from all of the sequencing efforts in prostate cancer. And indeed, these sequencing efforts have shown that there are numerous genomic alterations that are frequent in castrate-resistant prostate cancers, although they do not seem to be able to clearly classify the disease in therapeutically relevant subsets, with the exception of the DNA repair genetic defects. There has been some data that's shown that indeed up to 10%, perhaps a little bit of a lower percentage there, of men with metastatic prostate cancers will have germline mutations in DNA repair genes, and that up to 20% of men with metastatic prostate cancer will have somatic defects in DNA repair genes. And of course, these defects have been associated, as in other tumors, with sensitivities to PARP inhibitors and also to platinum agents. Now, a lot of work needs to be done here as well, because what constitutes DNA repair gene seems to be, really up to the investigator reporting the sequence. There are some that are more common, but I guess the denominator of the DNA repair genes is always in flux. And for some studies, the denominator is 130 genes, and for other studies, the denominator of DNA repair genes as 10 genes. And so obviously the percentage of actual alterations is varies based on that. And as with the FOXA1 gene, and as what we're proposing with the aggressive variant molecular profile genes, the details of the actual mutations that result in defects in these DNA repair genes need to be really determined. And not only that, as you can see here, these genetic defects all often happen in combination, so they're not exclusive of each other. And so what does that mean with regards to the functional DNA repair pathways? Moreover, the androgen receptor axis has been linked to DNA repair functional deficiencies. So how these DNA repair alterations impact androgen responsive and androgen indifferent diseases may in fact be different. And so based on the work of Tim Thompson in our group, who has demonstrated that the aggressive variant models that he's worked with are characterized by functional deficiencies in these DNA repaired pathways. We're now almost completing a clinical trial in which only patients with aggressive variant cancers, so now we're selecting for patients with only aggressive variant cancers, get treated with combination platinum chemotherapy and then get randomized to either observation or olaparib maintenance. And I have to say that the experience to date does indeed suggest that men that are treated with maintenance olaparib have a very prolonged or have a more prolonged progression-free survival than those in observation. And hopefully next year we'll be able to report on these studies, since we'll be analyzing biopsies obtained at baseline and biopsies obtained after the 6 cycles of chemotherapy. And then finally, the other thing that we're very interested in and that really needs to be elucidated is how do we link these aggressive variant tumors to immunotherapy response profiles? And I did not detail all our findings here. Our clinical experience in the numerous immunotherapy trials that are being conducted by Pam Sharma and Sumit Subudi in our group for men with prostate cancer, that those that have these aggressive variant features actually do very poorly with the immunotherapies. There's a sort of a joke going around in the department that, Chris Logostita's patients always do great on these trials and mine do not. We really need to think very carefully about how we select patients for these studies. And as you can see in the gene expression profiles that I showed you earlier, those patients that are not characterized by the androgen response profiles, that are enriched in the EMTs pathways, also have enrichment of a number of immune pathways such as IL-6, JAK-STAT3, interferon gamma response, TNF alpha signaling, IL-2, STAT5, etc., which could have been linked to possible, immunosuppressive microenvironments. And so we're doing a lot of work now with the IMT, Pam, and Sumit Subudi to really understand what the immune profiles of the aggressive variant tumors are and what are their immunotherapy vulnerabilities. And so, in conclusion, I hope that I have convinced you that prostate cancer is a heterogeneous disease and that this heterogeneity has to be taken into account in clinical and preclinical studies. So when anybody is reporting any finding in prostate cancer, I think it's critical that the androgen the androgen signaling responsiveness of the model and or the novel target or novel drug be studied in the context of the sensitivity to androgen signaling inhibitors. And so far, we've provided a framework to study these androgen in different tumors and to try to develop therapies that are specific for them by defining the aggressive variant prostate cancers. Although, of course, as I mentioned, a lot of work remains to be done so that we can make greater advances. And I think with that, I'm getting all these. I would like to point out that this is where we would like to go to a biologically based, clinically meaningful classification of prostate cancer that can accelerate our discoveries and clinical research advancements. And with that, I'd like to thank you for your attention and of course say that it takes a village. There are numerous people that have helped with all of this work. I probably missed some of them on this slide, and if I did, I apologize. But it has really been a whole village effort to conduct all of this work. And with that, I'd take any questions.
Thank you, Dr. Parisio, for this wonderful presentation, very detailed, extremely interesting. I have some questions from the audience. The first one is a comment and a question. The first comment is very nice talk. And then the question is, are there cases of classic prostate cancer shifting to androgen indifference upon tumor relapse, possibly as the result of cancer stem cell plasticity?
Yes, so there, as I mentioned in one of my earlier slides, a mechanism of secondary resistance to the androgen signaling inhibitors is the emergence of these androgen-indifferent tumors. Now, the mechanism by which those emerge is unknown. It appears that there is, so most of the studies suggest that there's a transdifferentiation of the luminal cells, or sort of the original luminal components of these prostate tumors, and that it is rather than sort of a clonal selection or a sort of a different tumor type emerging altogether. It appears to be a transdifferentiation. So there's this concept, which obviously is not exclusive to prostate cancer, of lineage plasticity that is being looked at, that is thought to be driven by epigenetic mechanisms. And so there's a whole effort to try to reverse this lineage plasticity and to reinduce responsiveness to androgen signaling inhibitors.
Thank you. So the second part of this question was, in other words, can some aggressive variant prostate cancer derive from androgen dependent prostate cancer?
Yes, absolutely. Yes.
And then I have another question that is, did you look at the OXPHOS genes in the ROS generating pathway in aggressive variant prostate cancer?
Not yet, but that's another avenue that we're starting to explore, is the metabolism of these, or the unique metabolic features of these tumors. But no, not yet. We haven't looked.
And then there is a third question is, did the rate of aggressive variant prostate cancer increase due to the increasing knowledge and recognition of this entity?
I'm sorry, can you repeat that question?
Is the rate of patients with aggressive variant prostate cancer as it increased thanks to the characterization of the biology and the recognition of the entity?
Yes, yes, and so what's happened in prostate cancer is an interesting thing. So initially, all we ever had was, it all started with orchiectomies. You took away testosterone, majority of prostate cancer shrank. That was it. And then we had the LHRH agonists and antagonists, which again took away the testicular testosterone. 30 years ago, if a cancer progressed, had castrate-resistant progression, we called it androgen independent. And for years, it was considered androgen independent. But then the scientific community, science, told us, and actually also in the clinic, it was seen that some agents such as ketoconazole that suppress adrenal androgens still had an effect in these patients. But between that, the clinical and the basic science, it was found that, in fact, these the mechanisms of resistance of castration resistance were still very much driven by the AR in a lot of the prostate cancer. So then nobody talked about androgen independence at that point. And now, in the last, I guess, 5-10 years, we've gone back to saying, okay, the majority of castrate-resistant prostate cancers are probably still driven by AR, but still, there's still a subset in there that is truly androgen independent. So whether these androgens, so there's a proportion of them, as I showed at the beginning, that are probably androgen independent from the get-go. And then there's a proportion of them that become androgen independent. Now, what makes them become androgen dependent is not clear. Was that program there to begin with, and it just sort of is unsurfaced or surfaces because of the androgen signaling inhibition? We don't know.
Thank you. So you think there is some possibility that there is clonal heterogeneity and the different clones can be present in different sites according to the evolution of the disease?
Yes.
So thank you. We have
Alessandra, one more question. Anna, it's a very nice presentation. Congratulations. So having this mutation in DNA repair gene, did someone look for additional mutation, let's say in enhancer DNAs, in super enhancer, in DNA non-coding region outside the exons in this peculiar type of prostate cancer?
Not yet, but that's work that we're actively doing right now, because you're absolutely right. If the genomic sequences don't explain these distinctions, then we're looking at the chromatin structure in the non-coding regions. Absolutely, we haven't looked yet. We're looking.
Thank you. So we are going toward the end of this session. I would like to thank you, Alison Gart and Elizabeth Kent, for their scientific support and assistance. And I would like to invite everyone to come back next week. We have 3 wonderful speakers that will be presenting next week. And then we will also be able to continue the series in a less frequent format past the end of May. So thank you everyone for attending, and have a safe and enjoyable weekend.
Thank you very much. Thanks.